Okay, Bio 201. This is the end of Chapter 9, what we did not finish on Wednesday, and this is your lecture, if you choose to make use of it, that I'm going to give to you in absentia. So I will be gone, of course, to the Kentucky Academy of Science meeting on Friday, but here's a brief overview of what we didn't cover in Chapter 9. So we finished talking about mitosis, and the only thing we hadn't talked about was the small part of the chapter at the very end that relates to cancer. And we had mentioned previously a little bit about cancer. We'd said that cancer is the result of a mutation in the cell, typically in a tumor suppressor gene or possibly in a different type of gene that we'll talk about here in just a moment. We said that the loss of control of the cell cycle leads to this thing we refer to as a tumor. We categorize tumors in two different categories, at least broadly. One would be, the first listed on our slide, would be a benign tumor. Benign tumors are not cancerous in that they don't invade surrounding tissue. They're typically encapsulated, and they're relatively easy to deal with because they remain in one place and can be removed. The second type of tumor is much more insidious and dangerous and difficult to deal with, and so that's a malignant tumor. And so malignant tumors are not encapsulated, and they invade neighboring tissues. So a benign tumor doesn't push too hard, or it doesn't start to move into surrounding tissues, but a malignant tumor does invade surrounding tissues, and worst of all can undergo this process referred to as metastasis. And as you're likely aware, metastasis occurs when a bit of the tumor breaks off, moves through the body, most commonly through the lymphatic system, reattaches in another place in the body, perhaps in a lymph node, and a second tumor begins from the cells that left that first tumor. So what we notice is that the development of cancer tends to be gradual, simply because it may often require more than one mutation and may take a number of years before a cell is obviously cancerous. Okay, we move to the next slide here. There we go. And furthermore, we want to talk a bit now about cancer cells versus normal cells. And this is appropriate given that you just finished your HELA cells in lab, and it's a little disturbing as well, to be perfectly honest. So if we look at cancer cells, we see that cancer cells are non-differentiated. So they are cells that possess your DNA, but that DNA may have an abnormal chromosome number and an abnormal nuclei, like we see in number 2 there, and the cells become undifferentiated. They do not do anything useful for the body like your normal differentiated cells. Now they do not undergo apoptosis, which is not surprising based on our discussion on Wednesday. Normal cells, of course, do undergo apoptosis, and then as we touched on a moment ago, normal cells have contact inhibition. So if they start pushing on another tissue in the body, they stop dividing. Cancer cells do not have that. Normal cells are typically organized in multiple layers. So there's one layer, like your skin, for instance. There's 30 layers perhaps in the epidermis, the stratum basale, the stratum corneum, and all those layers in between. And so those cells are organized into these very distinct layers. Cancer cells are not organized. They are completely in this disorganized array, and they divide but do nothing useful for the body. And then last of all, cancer cells undergo metastasis, like we mentioned, and unfortunately angiogenesis. Angiogenesis is the process by which cancer cells secrete signaling factors that cause new blood vessels to develop that deliver blood and therefore oxygen and nutrients to the tumor itself. And so some anti-cancer treatments focus on preventing angiogenesis, which slows, if not prevents entirely, the development of the tumor. So cancer cells are certainly not like normal cells. And now, perhaps most importantly, we talk here at the very end a bit about the genes involved in the development of cancer. So we have two genes that are most frequently involved, referred to as oncogenes and tumor suppressor genes. And so an oncogene is this. it's kind of an interesting description. An oncogene starts out as a proto-oncogene, which is a gene that produces some sort of product that promotes the cell cycle. Something like E2F, like we talked about on Wednesday. And so the catch is that a proto-oncogene promotes division of the cell, in other words, continuation of the cell cycle, at an appropriate rate and an appropriate number of divisions. And so if a proto-oncogene becomes mutated, it turns into an oncogene, and an oncogene causes the cell to go crazy as far as the progression of the cell cycle goes and to divide much more rapidly than it should. It's like I've seen analogies in textbooks related to pushing the accelerator down to, say, to go 25 miles an hour on a city street. That would be an oncogene. That would be a proto-oncogene. An oncogene is just mashing the accelerator down and flying, going way faster than you should, much faster than you should in a particular area. Perhaps that's a good analogy to illustrate the role of an onco versus a proto-oncogene. The second type of gene involved in the development of cancer is that tumor suppressor gene that we discussed on Wednesday. So P53 is the primary example. There are other tumor suppressor genes, but P53 is the most common example, simply because it's involved unfortunately, the development of so many different types of human cancers. As we mentioned, over half of all human cancers are thought to be tied to P53, believe it or not. So I think we got a pretty good idea of that on Wednesday. So it is, I think, probably self-evident that if a tumor suppressor gene like P53 has a mutation, it's unable to interact with DNA in the way that it should and perform this function of either fixing a mutation or leading to apoptosis, as we mentioned on Wednesday. And so, of course, when that happens, then a cell that already has a problem and is starting to divide too quickly isn't eliminated from the body, but rather is allowed to continue to divide and develop into a tumor. All right, guys, so last of all, we talked briefly here about prokaryotic cell division, and very little about prokaryotic cell division. Now, a couple differences. It's not referred to as apoptosis. It's not mitosis is what I meant to say. It's not mitosis, but it is what's referred to as binary fission. Prokaryotic cells divide in a process referred to as binary fission. And there's a couple key differences between bacteria and, let's say prokaryotic and eukaryotic cells that you're familiar with. Of course, the bacterial chromosome is a big loop, and it's 1,000 times or so the length of the cell, but it's folded up into this area referred to as the nucleoid. And interestingly, what we see is that this whole ring, as you would expect, is replicated prior to cell division, and these two replicated chromosomes attach to the plasma membrane. The plasma membrane splits, and we end up with two daughter cells that are identical. This is accomplished, eukaryotic cell division proceeds rapidly, but this is accomplished extremely quickly. E. coli going full tilt can go perhaps 15 minutes from one cell to two cells if they have everything they need as far as nutrients. Apparently, that in order to do that, they have to have a head start on that replication of the actual chromosome, so that the chromosome there are multiple copies of the chromosome that are being replicated, and then they just wrap one up every 15 minutes. Okay, and so our last slide really offers little new, but it does have a nice picture of this process of binary fission in prokaryotes. And so you can clearly see that chromosome replicating, and then you can see the cytokinesis splitting there as occurs in bacterial cells. So, binary fission, of course, is a form of asexual reproduction. There's no scrambling of genes here. As we've talked about, bacteria can do that via conjugation, in a manner of speaking, but binary fission results in two cells that are identical to the parent cell. Okay, so this finishes up Chapter 9. We did almost all of Chapter 9 on Wednesday, but there's this little bit here at the end, and so that's a wrap for us for Chapter 9 for fall of 2017.